our next speech, we'll be hearing from Marie Lamar, who I know is actually here, and who is honing her presentation mastery by working on her vocal variety and body language. She's a Science and Technology Policy Fellow at the National Science Foundation, who supports the Research Experience and Mentoring Program, helping to provide more opportunity for underrepresented groups in engineering fields. So please welcome Marie Lamar.
Thank you, Mr. Toast Leader. Okay, I will be talking about biology again today. I'm going to start with the basics. How many of you have heard of a cell? I'm not talking about a prison cell, but a biological cell. Cells are the building blocks of life. All of the organs and tissues in our body are made up of cells. Some of the most famous cells I'm sure you've heard of are the sperm and the egg. If they get together and start dividing into many cells, it becomes an embryo. But when scientists want to study biological processes that happen in human cells, we likely can't get access to things like embryos or live tissues. So we use something called cell lines instead. A cell line is a group of cells, usually derived from a single cell, that you can grow indefinitely in a petri dish in the lab. Cell lines are an amazing resource for scientists. We can use them to study human development, genetics, disease mechanisms, all sorts of things. In my postdoctoral research, I use cell lines derived from patients with epilepsy to study differences in gene expression and neurodevelopment using 3D cell models called brain organoids. I also needed cell lines from healthy controls for comparison. I made sure to obtain appropriate control cell lines that were matched for age, sex, and ethnicity. It's important to have appropriately matched control lines because biological variables like age, sex, and ethnicity can cause differences in gene expression between cell lines. In 2014, the National Institutes of Health, or NIH, released guidance about accounting for sex as a biological variable in cell lines. However, other factors such as ethnicity currently do not need to be reported or considered by researchers using cell lines. The vast majority of cell lines used in biomedical research are of European descent. This lack of ancestral diversity in cell lines used for biomedical research leads to research findings that are not applicable to much of the population, increasing health disparities that already exist for underrepresented groups. Therefore, I recommend that NIH have gumption and start to require grant applications to consider cell line ancestry as a biological variable and require investigators to report the ancestry of the cell lines used in publications. Although socioeconomic factors play a big role in the health disparities seen between people of European descent and people of color, there is evidence that genetic ancestry also contributes to health outcomes. For example, Black men have an increased incidence and mortality rate of prostate cancer compared to men of European descent. And researchers have found that different genes are commonly mutated in cancer cell lines from white men. Cancer cell lines from Black men compared to cancer cell lines from white men. These genetic differences can explain some of the disparities in prostate cancer risk and aggressiveness between Black men and white men, but also have big implications for precision medicine. Researchers use commonly mutated genes in cancer tumors to then identify pathways to target with drugs. And if researchers only had data from cell lines of European descent, they would target these genes that are not as commonly mutated in Black men, failing this population. Pharmacogenomics, or the use of genetic information to predict the drug response of an individual, is one of the core elements of precision medicine, where medical treatment is tailored to the individual needs of a patient. Pharmacogenomics data largely comes from research on cell lines with different genetic backgrounds. When researchers perform drug efficacy or toxicology studies on cell lines that are not ancestrally diverse, this robs a big part of the population from the benefits of precision medicine. An example of this is the case of the HIV drug Sustiva, which depends on a specific enzyme to be metabolized. Africans have a high frequency of a loss of function variant in the gene that encodes for this enzyme, leading to high toxicity and a higher frequency of adverse drug events in this population. Had researchers used ancestrally diverse cell lines to study drug toxicity, they could have recommended lower doses for Africans carrying this loss of function variant. If researchers were required to report the genetic ancestry of the cell lines used in their studies, this would have an added benefit of increasing the integrity and reproducibility in research, because researchers would have to confirm the identity of their cell line through genotyping prior to publishing. Believe it or not, this is a big problem, because studies have found that up to 36% of cell lines used in research are either contaminated or incorrectly labeled. One barrier to implementing the proposed policy is that biobanks are lacking in cell lines from ancestrally diverse populations. This is in part due to the mistrust of the medical research community among people of color, due to historic exploitation and abuse. So medical researchers must work to build back trust in minoritized communities to obtain these vital samples. Researchers may also push back on this policy because it will impact their research budgets, as well as increase time spent on genotyping when there is pressure to publish quickly. However, the advantages far outweigh the risks, as we expect the result of this policy will be increased accessibility of precision medicine for diverse populations, a better understanding of health disparities, and an increased reproducibility and integrity in research. Thank you.